Hi everybody, I wanted to welcome you to Lecture 4, where we're going to be talking about some of the morphology of the cortex and of fibers of passage. We'll be looking at some of these different types of association fibers that help to connect different regions of the hemisphere. We'll talk about projection fibers and commissural fibers, and how the wiring works within different regions of the brain. So the first thing we're going to kind of work to kind of tune into here is this differentiation between white matter and gray matter. And if we zoom in a little bit, we can see how we have this major division of these two different types of tissue within the brain itself. And what this reflects is, as we may remember from earlier conversations about the development of the brain, is that this outer region is where we have lots of cell bodies of the brain, right? These are all neurons with their cell bodies. But this white area is white because it's relatively fatty. It has myelination, it's myelinated sheaths. These are oligodendrocytes have wrapped around the axons of neurons that could reside here, for example, and projecting down through here, or and these projections that arise from these cell bodies are myelinated. So what we have then is that we have different types of fibers of passage then that help to connect between the different hemispheres, connect within a hemisphere, and also ones that arise here and move downward. So this type of complex connection and this white and gray matter that we see here is part of a larger story that we can that's about how these cells arise and how they're organized. So again, we're talking about how gray matter and white matter is divided, and what I mentioned before, cell bodies and dendrites. And I didn't bring up dendrites there, but those are where the input part of the neurons. And then we're in the white matter where we have axons. So okay, so what is it? I mentioned in passing what actually causes this kind of myelination. Oligodendrocytes form the myelin within the central nervous system. And if we zoom in here, we can see how the oligodendrocytes are, oligodendrocytes help to form the myelin within the CNS. And so this is a cell body of an oligodendrocyte that's wrapping around multiple different axons within the central nervous system. That's a key characteristic. And also kind of unique, because in the peripheral nervous system, we also have a myelinating glial cell. It's known as a Schwann cell. It's different because one Schwann cell wraps around each of the different internodes, that myelinated internodes between the nodes of Ranvier that make up an axon that we have within along the neuron itself. So Schwann cells are in the periphery, important to remember. Oligodendrocytes are in the central nervous system, and then what we have are astrocytes. And so astrocytes are present within the central nervous system, and they help to form, provide buffering for the neurons as they fire and so forth. They create a lot of potassium and excess amount of metabolic activity, pH changes, and the like. And so they help to buffer and control that. They also play really critical roles in being able to help maintain the homeostasis of neurons, so that they're able to persist. In addition, they also help to form these end foots along capillaries themselves. So there are other types of glia as well, known as radioglia, that are similar to astrocytes and that are responsible for making and serve as progenitors early in development. In fact, if we look at the development of the central nervous system, we'll start to see these radioglia and how important they are in the functioning of the nervous system. So this very early stage here, this is basically an early embryo that's been cracked open. So what we're looking at is the interior sort of ventricles or vesicles that are part of that early part of like the telencephalon that we have right in here. And if we look at this and zoom in on this particular area, we would see a single cell layer containing these early neural progenitors or neural stem cells. And so this is what they describe as being this sort of single layer that's important here, right along the surface of the nervous system, along the surface on the interior of these vesicles here, is what's known as the ventricular zone, and that persists for a long period of time during development. and is responsible for and helps and houses many of the different stem cells that help to create the constituents that reside within the developing nervous system. So if we look at the development of the cerebral cortex, we end up getting 6 layers, including a white matter layer in the adult, which we can see out here. And how that actually ends up arising is that the ventricular zone, very early in development, starts going through a series of divisions and gives rise to these multiple different plates, like the preplate region that we see here at this stage here, which is in humans between weeks 8 to 9. Then as time goes on, the next 2 weeks, the weeks of 10 to 18, then we get more and more of this expansion of what's known as the cortical plate. And this helps to split that preplate into the marginal zone and the subplate region, which is down in here. This cortical plate, what ends up happening is you can see, and I'll kind of zoom in here, is that we have these kind of bluish kind of shaped cells here, and these are called radioglia. And then riding along here are the neurons that are that ultimately will move outwards and then peel off of the radioglia and reside within the cortical plate. And so what ends up happening then is that you have this kind of inside-out type of appearance of cells that start to arise here, so that you end up having these different regions forming these 6 different plates or layers. Okay, fine. So when we're looking at this, then we can see this is a depiction again of that radial process of a glial cell, and then this sort of rainbow-colored migrating neuron that moves outwards here. And so they're constantly moving through the cortical plate and dropping off here, and sort of adding on new cells on these outer walls here. So these multiple different layers then arise at different times during development, and they also have different types of functions that are associated with them. And so Ramon and Cajal looked through here using a specialized stain to be able to characterize some of the morphology of these different types of cells, and basically found that of these different layers here, he could see some morphological characteristics to try to help describe them. So in order to be able to appreciate what some of the functions are and the organization of some of those different types of cells, what we can see here is this general classification of fibers that we have. Those bundles of white myelinated axons that we talked about before. So this simple schematic is basically illustrating how within each of the different hemispheres we can have fibers that project from maybe one part of the cortex to the other, and those would be associational. We also have fibers in which neurons that may have their cell bodies on one hemisphere project over into the next hemisphere, and those are described as commissure, and they're crossing the commissure. Then what we also have here is projectional. These are ones that arise in the cerebral hemisphere and then project outwards and either into the bulbar or a spinal cord region and synapse onto different neurons, sometimes directly onto the lower motor neurons themselves and other times onto interneurons that precede synapsing onto the lower motor neuron. Interestingly, many of these different projection neurons, if they go past the level of the medulla, will decussate or cross over. We'll talk more about that in a later lecture. For now, we want to be able to appreciate that we have those different types of fibers of passage, and that illustrates what these neurons end up doing that reside within these different layers. So we have lots of cell bodies here, and then as we talked about, we have these projections that arise from those cell bodies. So how do these things actually end up connecting? If we look here, we see that these different the numbers that we have listed here are recapitulated here, and the major ones that we want to be able to appreciate is layers 4 and 5, but we need to know them all. So layer 1 is a most superficial layer, lots of synaptic connections, but lacks the neurons themselves. Layer 2 contains small neurons, and these are intracortical, so they have association and commissural fibers, so that's important. And so these are about trying to connect one part of the cortex to the other part of the cortex. Likewise, then, in layer 3, we also have associational and commissural fibers, but these contain slightly bigger neurons, these medium-sized neurons. Layer 4, highlighted for you here, is basically the site of termination of thalamocortical fibers. What that means is that these fibers are coming in from the thalamus, that deep nucleus within the brain, and these are carrying often sensory information, somatosensory information, or visual information, or auditory information. The thalamus serves as this relay between where we get input coming in and through some kind of sensory fiber coming in. It synapses within the thalamus itself, and then from the thalamus it projects to the cortex. And those thalamocortical fibers end up terminating in this layer 4. So if you're looking for a rich area where you have lots of a really thick layer 4, it's likely that that is going to be a sensory cortex. Layer 5 contains giant pyramidal neurons, and that give rise to these projection fibers. And so depending on which part of the brain you're looking at, they can be extremely large. So they can project to the spinal cord, for example, and other areas. But in layer 5, you can see this even in this simple depiction here, where we have them, and they're much larger here, have a pyramidal type of shape, and very long and large projection patterns. So that's layer 5. So this is all about output, going out to the basal ganglia, those deep structures within the cortex, within the hemispheres themselves, the brain stem, and the spinal cord. Whereas here in layer 4, it was a lot more about inputs and what was going on with different types of inputs. Okay, so we described these different layers. We talked about how they emerged during development, and then we talked about some of the constituents within there, talking about just the division of white and gray matter, how different glia arise, and so forth. Let's go into a specific example of some of these different types of cells that we have here. So in the precentral gyrus, this is where we have the primary motor cortex. So Brodmann's area 4 is where we have these here. And what's really interesting is that if you zoom in and you look closely here, these dark spots are large Betz cells that reside within layer 5 of this area here. And so this layer is really predominant in this region here. Contrast that to the other side of the central sulcus. Over here we have the somatosensory cortex on the post-central gyrus. And as you know from lecture 3, this post-central gyrus has many has Brodmann's areas 1, 2, and 3 in there. And if you zoom in here, you can see some of these types of granular cells that we have that are stellate types of cells that they have in here. And so these are the ones that we typically we see within the sensory cortex. So layer 4 is abundant in this region here, and layer 5 is really abundant in this region here. So remarkable that this type of function that we see, that we associate with these different types of regions, can be actually identified and guessed at just by looking at which layer is actually expanded and knowing what the basic function is of those types of layers. So again, we're looking at these different types of fibers that we have here: commissural, associational, projectional, and so forth. And what we want to appreciate now and dig into a little bit is talking about how we have these different types of fibers of passage here. So we have these association, commissural, and projection fibers. And what we have are some of these major types of regions here. The corpus callosum is one that you'll see a lot of. It's really easy to identify in some of the gross sections of the brain. And we can see how we connect one hemisphere over here to the next hemisphere over here. And so, and that's in contrast to what we have for associational fibers here shown in blue that help to connect within the hemisphere. So short associational fibers, like the ones that synapse in layers 2 and 3. And then what we have down here is we have some of these fasciculi, superior longitudinal and inferior longitudinal fasciculus. So these types of fasciculi project anteriorly and posteriorly, and we'll talk about some of those in the function in a bit. So, and this particular area here, shown in red, are white fibers that project through these different deep nuclei. So the basal ganglia are within here, and we talked about how the caudate nucleus makes that lateral wall, the lateral ventricle here, and some of the putamen and the globus pallidus that we have here. Well, this reddish part is described as being the internal capsule. And it's this capsule, sort of bundled bunch of fibers of passage that pass through here, and they're on their way down to either the bulbar region, that midbrain, or sorry, the hindbrain medulla region, or else they're down, projecting on further down to the spinal cord. And some of these are going to the midbrain as well. So, commissural fibers. Here we can see them. Corpus callosum is made of it. So if we look at the corpus callosum and somebody asked you, hey, where are these fibers going? Are they? You can say, well, they're commissural fibers, and they're connecting the two hemispheres. So they're fibers of passage that are going through, that are moving through here. So that's one of the basic kinds of characteristics that we can see. We can also see this anterior commissure here that's helping to connect the two hemispheres as well. So if we take a sagittal section and look at what's happening and just chip away at the brain that way, we can see where we have the corpus callosum here, where we've hacked across, and we've been able to see we've cut through that corpus callosum. Okay, awesome. So, and here we can actually name some of the different structures here. So what we have then is we have the rostrum at the very front. Think of the rostrum in a room, or rostrum as part of sometimes where they have the place where a speaker is particularly talking. Genu, which means bend. Body, which is this main region here. And splenium, which is in this back area here. And so these are some of the major divisions that we have of the corpus callosum here. Now, where these actually break off, like where the body is versus the splenium, we'll be sure that the arrows are pointing in really obvious areas if we ask questions about these different domains. Okay, so fine. And then what we're looking at here is that we have part of the anterior commissure as well that we've cut through here. So this cut through here is helping to connect the two temporal lobes to each other so that they can better be able to communicate with each other. And this is a really nice sort of allows that communication to occur for different types of auditory input and the like. All right, so what about these different types of associational fibers that we have here? What's going on with those? So these are kind of cool as well. And what we're looking at here is we have, if we look from the side of the brain here, we see these different types of fasciculi. So these pictures here are drawn from that McNeil book that's saved as a PDF on Blackboard for you to use as a study tool. And what we have here is this superior longitudinal fasciculus, sort of depicting the connection then that we have of the most posterior portions of the brain with the most anterior portions of the brain. And so this sort of superior longitudinal, or sometimes called arcuate fasciculus, especially in this area here, is important for, it just sweeps in this great arc over the what they describe as the insula of the frontal lobe, and goes back to the posterior portions of the hemisphere. So these help to provide some very strong types of communications between those different domains. Likewise, if we're looking at this inferior occipitofrontal fasciculus here that runs in this domain here. So these pass below the insula from the frontal lobe and through the temporal lobe and back into the occipital lobe. And its fibers fan out at both ends of the fasciculus here. And then the unguate fasciculus. This is actually a really cool one that if you lesion, you can really affect different types of memory recall. Because much memory processing happens here within the temporal lobe. And then this frontal lobe here has a difficulty actually being able to process that and make associations with it if those fibers are actually cut. So these help to interconnect the anterior and inferior parts of the frontal cortex, and also the anterior part of the temporal cortex. So if we look at these other domains that we have here, I'm looking at the sort of the medial side of the cerebral cortex here. We have what's known as this the cingulum. And so it starts here and moves around and ends here. And so it's moving around this parahippocampal gyrus that we have down here in the temporal lobe, and is important for helping to be able to function in limbic function and connecting the types of functions that are important for emotional processing within this particular type of region here. And so it's connecting some of the frontal part of the brain that's important in that kind of processing and context with some of the more primitive portions of the brain that are located in the temporal cortex. So, fine. We see these different types of fibers, and then arcuate fibers, which help to connect different domains that we have within the different lobes themselves. For example, up here in the parietal lobe. All right. So, if we're considering these different types of regions and how they work, then what we've seen is that if you remember that superior longitudinal fasciculus, which can also comprise of the arcuate fasciculus, this is one that helps to connect the frontal lobe with the temporal lobe. So to go back, we're talking about this portion of it right along in here. And so this arcuate fasciculus shown in this domain, from this perspective, looking down on the brain, we can see it's here, it's labeled A, right along in here. And then this is Broca's area, and then this is Wernicke's area. And so these different domains of the cerebral cortex are linked together. So we want to be able to name and number the different Brodmann's areas that Broca's area comprises of, and likewise with Wernicke's area. What are the numbers that are associated with that we learned about last time in lecture 3? We can see these different types of fibers in several ways. One of them is that we can see it in how the fibers are coursing down along the brain and down outwards, and how they arise within the cerebral cortex up here, and then how they project downward, almost like a funnel, and reaching down and finally getting down to the spinal cord itself. And so the fibers, if you peel away and just look at the fibers of passage along here, you can see how they're bundled together and they have a very organized type of structure here that ultimately then leads to and synapsing with some of these lower regions of the central nervous system. So these types of projection fibers that help make up this are known as the coronal radiata, and so they form they converge and form this curved band of white matter known as the internal capsule. And so what we're looking at then is this, and the internal capsule then ultimately then feeds into what's known as the crus cerebri of the midbrain, right down in here. So this coronal radiata and internal capsule, how are we able to visualize how these different structures work, and then what does this actually reveal to us about how the brain's actually put together? So we're going to get into these different types of projectional fibers for this reason. So we'll talk about the corticospinal and thalamocortical tract. So these are ones corticospinal, connecting from the cortex down to the spinal cord, and then the thalamocortical tract, which is connecting the thalamus up to the cerebral cortex. So corticospinal is an example of an output kind of projectional fiber. And then thalamocortical are these input fibers. So we can think of them actually synapsing into layer 4 of the cerebral cortex. And we can keep in mind what we have is this really nice picture that we have here of these projection fibers that are coursing downward through the midbrain on their way out to synapse onto the spinal cord. And so these are examples of these kinds of projection fibers, corticospinal tracts. Okay. And so this idea of basically how we have a thalamus that's taking in sensory input here and then passing it on up to the cortex through thalamocortical inputs, or how we have the cortex then sending out projection fibers then to ultimately project into the spinal cord in this type of example, is shown in this kind of simplified schematic. And the idea of it is to help you visualize and appreciate that we have a unidirectional type of association with these fibers. They're either going north or south, if you want to think of it that way. You don't have a mix-up in terms of which way traffic is flowing. And so sensory inputs are flowing upwards and towards the brain. So thalamocortical, for example, is an example of the sensory type of input, whereas we have motor output going around in this way. So, okay. In our next series, what we're going to dig into is getting into that internal capsule. But let's take a breather.